In the back. Okay. Hello and welcome to the Director Seminar. Thank you all of you for joining us today. Thank you to people on Zoom. Can't see me with you, but I am. I'm Marianne Alleyne, Director of the Beckman Institute. Okay, before we get started, I want to invite everyone to a special event, people online as well. Pay attention, we have pumpkin painting on Tuesday, October 25th. This is supposed to be a fun event. It is a creative wellness activity, so you should consider it as an important part of your workday to get your creative juices flowing and de-stress your minds to be more productive researchers in the long run. So everyone's welcome, faculty, students, staff, no artistic ability necessary. Seriously, you can finger paint on your pumpkins. I guarantee that you do that for an hour and you'll just feel happier at the end. You do need to register online, so go to the events page on the Beckman website or visit go.beckman.illinois.edu/pumpkin. So sign up, we provide everything. We provide pumpkins, we provide paint, we provide the fun. You should do it again. You know, the more people who come, the more we'll do activities like this. It will make all of us happier and better, and that's the goal. Okay, so with that, we have another special treat today. We have a seminar led by Professor Marianne Alleyne. She is an assistant professor of entomology at the University of Illinois, and she also holds affiliation in the Department of Mechanical Science and Engineering. Here at Beckman, she's part of the Neurotechnology for Memory and Cognition Working Group. Marianne received her BA in Integrated Biology from the University of California, Berkeley, her MS in Entomology from the University of California, Riverside, and her PhD in Entomology from the University of Illinois. She is the current president-elect for the Entomological Society of America, the largest organization in the world serving entomologists, and will serve as president in 2023, focusing on insect science policy and advocacy. So I don't know if you guys knew this, but there are 10 quintillion insects in the world. That's 10 to the 18. If you put them all together and put them on all the people, they will squash all of us. So think of that, be grateful there's no retribution here. And more importantly, they could be the key to understanding our biology in the world. So Marianne knows this. Her insects are the inspiration for her research group, the Alleyne Bioinspiration Collaborative, also known as the ABC Lab. In the ABC Lab, researchers from different fields come together to study a variety of insects, designing materials and mechanical systems based on insect biology. In today's presentation, Marianne will introduce a design framework developed and used in the ABC Lab. The circular framework enables interdisciplinary teams to uncover the fundamental physics and chemistry behind biological processes, uses that knowledge to design bio-inspired technology, and applies that technology to better understand the world around us. Her talk is entitled, Insects Did It First, Multifunctional Surfaces and Structures. Thank you, Marianne.
Thank you, Director Mason. Thank you. Should we turn that one off?
I turned it off.
Okay, thank you. All right. Am I all hooked up correctly? I think. Hope Zoom can hear me. Yep. Oh, I have to, there we go. All right. Thank you all for coming. Thank you for the invite. I am feel incredibly privileged that I'm part of the Beckman as an affiliate. It already has created great benefits to me in the last 3-4 years. I don't know how long it's been. It's time is not. It's hard to figure out where we are at. And I'm going to be presenting some of the stuff that is most related to the Beckman Institute today. So I want to introduce first what bioinspired design or bioinspiration or biomimicry is to me and to my lab. I'm going to talk a little bit very quickly about the ABC Lab projects, focusing on cicada wings and leafhoppers, brochosomes. I'll explain a little bit more about that later. Focus on some of these new tools that we use in entomology now, thanks to whatever I'm learning from you all here at the Beckman. I mean, we're using tools that have never been used in entomology. And then also because the Beckman Institute prides itself on interdisciplinary research, also want to discuss a little bit about what I've learned over the last 10 years or so about the benefits and challenges of that kind of research and where I think we can maybe make some changes. And then since this is not primarily an entomology crowd, I also want to end with saying a few things about insect biodiversity and global change. So what is bioinspired design? Well, you probably have heard of this before. These are probably the two most famous examples of bioinspired design, of bioinspiration. There is, so the lotus leaf is super hydrophobic. It makes it that water droplets sort of coalesce, and then they roll into the middle, cleaning up the leaf and making it very efficient for photosynthesis, more efficient for photosynthesis. And this has inspired paints, windshields, and so on. The other, probably most successful and thing that makes the most money is the one I can never pronounce, hook and loop fasteners. Okay, Velcro, which was discovered in the 1950s, and it basically was inspired by the hooks of a plant burr that keep getting attached to the hoops of dog hair. And so that was the inspiration for Velcro. Insects have also inspired many new technologies, and I wanted to show you sort of at different scales. So the top right is ants and ant behavior. It can be any kind of social insect. Also goes for honeybees. They have inspired new algorithms, new, more efficient ways of connecting, maybe how computers talk to each other, and so on. Many examples there. So this really you're looking at the whole community and how individuals interact. Then there's the example of morpho butterflies, which use structural color and to get this beautiful iridescent color, which have inspired improvements in displays. Termite mounts, like these big termite mounts you'll find in Africa and Australia, that are kind of porous at the top, but inside here is the nest and the fungus that termites farm to give to their offspring. And these have inspired buildings that don't need HVAC systems. So this is the Eastgate building in Harare, Zimbabwe, for instance. And then the other one is an insect that is inspired materials for collecting water. The Namib beetle, it stands in the desert, in the Namibian desert, and in the morning when the fog rolls in, it actually can collect water and drink from that water. It doesn't actually have to rain or find a puddle of water. So that's by having on the cuticle, which is the exoskeleton of the insect, super hydrophobic and hydrophilic patches. I'm also putting these up, not just because they're an example of insects, also to, I guess I'm getting on my soapbox here a little bit, to point out some of the problems with bioinspired design or biomimicry. These displays that have been created, inspired by butterflies, were sort of commercialized by Mirasol, which was, I forget what a subsidiary was from, but it worked really well, but just couldn't compete with other things that are on the market. So that's always an issue, right? So you might have a great idea, but if it's not competitive, then it's not gonna you're not gonna find it in your house. Here, the idea was that termites are really good at maintaining the same temperature in the nest, and they do this by this convective cooling that happens in the termite mound, and therefore we can also put that in buildings. Turns out if you do build buildings like that, you still need fans and stuff to run, because only later, like only maybe 10, 15 years ago, did actually people put a thermometer inside the nest. And it turns out that it's actually not very good at maintaining the same temperature. There are other reasons why they built this. Maybe ventilation, clean air, that kind of stuff. So I'm not saying that this is not a great building. It is. It costs a lot less money to build because it doesn't have an HVAC system. It is pretty cool during the day, and during the night they get rid of all the hot air. Actually, the U.S. Embassy is in this building. And then a word of warning is for the meat beetle. This is a great idea, but again, it turns out that the biology might not be quite correct. Behavior of the beetle is also very important, not just the cuticle. Also, where it stands, it usually is in a gully. So the way the wind blows is also really important. And then you also get these, like this is a great example. There's hydrophilic areas and hydrophobic areas in this fabricated material, very similar to the Namib beetle, even though I am not sure that picture is actually of a Namib beetle. That's a whole other thing. Like insect taxonomy is not usually the strong point. And then this one here, actually, this dew bank, it collects dew and then collects it during the night in the water here. Great idea, but they actually can't read this. But they say that they were inspired by the Namib beetle. I would argue this is nothing like the Namib beetle. So there's a lot of sort of greenwashing that's happening too, where just because it's bio-inspired, it's more sustainable. While Velcro is still made out of fossil, petroleum materials. So anyway, what I try to instill in my students is that bio-inspired design is all about building appropriate analogies. It is not just about copying everything exactly. And we just have to find those meaningful links between nature and technological innovations, which is actually not that easy. And but we recently, hold on. So we do this analogical reasoning that we're trying to develop we got some money from the Air Force, actually, from the FRL, to actually try to figure this out. Like, how do you define and retrieve what you're actually seeing in nature or what you are needing in your technology? Align that and abstract it between the two, and then also evaluate what you get and make it a reiterative process. I'll explain a little bit more about that in a little bit. So let's try that here. So we in the ABC lab, we look a lot at surfaces that have differing wettability characteristics. So that you can influence condensation, evaporation, fogging, droplet jumping, heat exchange, friction, and so on. So where do you think this might be applicable? Where do you think we could use surfaces like this? Glasses. So non-fogging glasses. Windshields. Oh, that's not a glass. Yeah. What's that?
Pins.
Pins. Okay, pins. Yeah, so actually the material. Okay. Anyone else?
Internal cameras. So surgeons are always talking about the cameras that they use for arthroscopic surgery, fogging up.
Okay.
Every single time.
Okay. Give me a call. All right. So yes, that's all. That's what we do. We look at self-cleaning materials is of interest, and anti-fogging, of course, of interest. Also, this is work I do with Nenad Milkovic and Mexi. He's of course very interested in anti-icing, but also we go a little bit further, making it a little bit more abstract. I maybe not quite, might not initially see the analogy between the two, but if you have a surface where water droplets can jump away from a hot surface, then maybe if you incorporate that in microchips, I mean, no equivalent in nature to microchips, but we can maybe make a bio-inspired surface there. Another one is HVAC heat exchangers to actually, again, if we get water droplets to jump and then be carried away, maybe we can make them more efficient. So it's not always that we're just creating new materials, it's also trying to really influence technologies. Okay, again, getting on my soapbox here. There are a couple of myths associated with bio-inspired design that I think kind of diminish sometimes the role of the biologist in the whole process. So many projects, a biologist will be involved in the beginning, and then we know everything about the biology, we're done, and the engineer just keeps going. Then it doesn't make it very attractive for biologists to be part of this collaboration, I would argue. So some of the myths that I've come across is that all knowledge necessary for inspiration is already published. It's out there. You just need to find it, you need to mine it, and then it's all there. You don't actually need even a biologist on your team. All biological discoveries are sufficiently abstracted that you could easily make these connections between what you need in your new technology and what's happening in biology. So biologists are, again, not needed to really understand the basic principles, but like the physical principles or the chemical principles that are working. All constraints that might invalidate these analogical reasonings are known. We know how we got here, how these systems, these materials came about, and that's good enough. We don't really need to know how they came about. Because organisms are optimally designed, and that is not correct. Okay, so that's I would argue that natural selection creates organisms that are just good enough, and just good enough pretty much yesterday, right? Like they're not optimally designed at all. And then all current engineering solutions are known to be inferior to biological solutions. I also think that's a myth, right? I think we could actually improve on nature if we really understood it. So all of these are wrong. I think we do need biologists and engineers to work together throughout the project and keep iterating. So this is what we do in my lab. So we try to understand biology, the insects. We explore the constraints and opportunities, try to figure out why something might have evolved and what were the constraints. Like, why do we not see, 8 legs on an insect or something like that? And then which makes the lab very different to a biology lab, I think, is to actually create optimal solutions, like actually prototype. Actually, that's not true. There's other labs that do that, but not that many. To actually create prototypes where the biologist is involved. So that would be one payoff, but then for the biologist, the payoff is also to go the other direction, to then say, create these materials, change a couple of things, and then help me explain why I'm not seeing this in nature, and it's the stuff that we couldn't do otherwise. All right, so here's another way of showing that. We use biology to learn from the biological solutions to improve engineering systems, materials. And we also can use engineering to apply the models that we're creating in engineering or the experimental tools to answer key biological questions. And so I'll give you some examples of that. Going back to insects, like you said, there's a ton of them, not just individuals, but also different kinds. Here are probably some that are familiar to you. Is there one that you really want to know what it is because you don't know what it is? This one? Yeah. That's a tick. It's not an insect, sorry. But we adopted all of pretty much the spiders and things. Here's another one. Then also think about
The clear one in the middle.
That's
Yeah,
right.
that one.
That's a termite. Worker termite. So, so also think about like all of these also have different forms, like a fly. This creates stuff as an adult, but also as a large, when it's just a magnet, it looks completely different. It has all the different structures. So it's just not, there's all these different, and they live in different niches, different habitats. And so there's a lot of inspiration here. So even in my lab, we study a lot of different insects. And it's sometimes kind of frustrating because many of these insects we can't actually rear in the lab. So you really dependent on your summer season. But we study these. I kind of have divided it into 3 different topics, even though Xavier, new student in the lab, is probably going to go a completely different direction. So we may have to add another. So we study primarily multifunctional materials. So that's what I want to talk about today. Even though the movement one, this is stuff that I did with Amy Wissa. You may have read the papers that we read. We wrote on that on the click beetle jumping, but I won't be able to get into that today. So multifunctional materials. And what do I mean by that? It's my materials that have different functions like wettability. They might be antimicrobial, which might be kind of related to each other. Iridescence, reflectivity, contact mechanics. They may have certain materials have certain kind of friction that will help the insect. And also this elastic behavior of the material is important. So we study all of this in all these different insects. So dragonflies, grasshoppers, flies, cicadas, leaf hoppers, click beetles, and other kinds of beetles. And when Tanya was in the lab, we also studied ticks, but they've been, sorry. So this is currently my lab. This was the first time we went out to dinner together after coming out of COVID. So everybody has their own, I mean, I couldn't be doing this without them. They're wonderful to work with. Okay, so again, multifunctionality of materials. This is a great paper by Schroeder. This is not Charles Schroeder, not our Schroeder, but I mean, actually, you should claim it. It's a really great paper. Just want to make the point. Insect cuticle or exoskeleton or the integument, I mean the same thing by it. Very simple building blocks. It's like a couple of sugars, the glucosamines, and it is proteins that bind together. Sometimes there's some metals incorporated, but that's only like at the jaws or something like that. It's very simple building blocks, but you can make all these different kinds of cuticles. So imagine, you probably all know the caterpillar, monarch caterpillar, nice and soft and squishy, but then the monarch itself can fly, it's pretty flexible. And then in between, there's actually, you might have never seen it or touched it. The chrysalis state is really tough and hard. All of that is the same material. It's just processed differently. So what we do is we then study all these different motifs and then the different functions. So we are kind of trying to figure it out where we're all playing. This is our playground. I wouldn't be surprised if eventually we cover everything. All right, so this first, this project I want to discuss is like I said, together with Nanad and Don Kropek at Searle. So this was also funded by Don, by the U.S. Army Corps of Engineers. So this has been a great project to get entomologists and engineers together. And then Don is a chemist. Okay. So what we started with studying Midwest species of cicadas. We knew that cicadas have nanopillars on their wings. At least that was described in a couple of species in Australia. So we wanted to see if that is here too, and if we actually can make predictions about where a species might occur what kind of nanostructures they might have. So something, this is the one that you see here all the time versus one that is in Southern Illinois in a really swampy habitat. This is my favorite. It's in prairie, which is not that common in Illinois. Therefore, this is not a very common insect, which brings all kinds of problems. And then we have these, what we call periodical cicadas, these ones that come out every 17 years. And then, because we are entomologists, we actually have names for each of these areas on a wing. Like this is the MC cell, this is the U3 cell. We actually would measure things like the microstructure, the nanostructure in these cells, and also the hydrophobicity characteristics in those cells. So, but we were very particular because we felt that if something is at the leading edge, it might be very different from the trailing edge. So, and this was, I mean, this is already a great step forward in this kind of literature. When we created a table of comparing it with previous studies, there were a lot of papers on the brown cicada from Indonesia or the black cicada from Australia. So names are important. We did 4 from Illinois. There are actually 20. Also, when we published this, somebody said, so why didn't you do all 20? Sure, if you give us the money, we'll do 20. So this is what it looks like. So this is an SEM done here at the microscopy suite. From viewing from the top, they're beautifully arranged nanopillars. And they're sort of like hexagonally close packed structures. Like here's the hexagon, and just. I mean, what are we looking at? So this is if you put the wing of a cicada under an SEM and you look straight down, you just get the tops of these nanopillars, and then someone here is a little bit at an angle, so you can see that it is a nanopillar. It's within one segment. Yeah, so this is in the MC cell. Okay. And then we compared all the different cells together. We also did AFM. So here, this is what that then looks like. So certain species have really nicely defined nanopillars, whereas other species, this one, and do you remember which one that is? It's the one, it's the actually the periodical cicada. So it is the cicada that only lives 2 weeks as an adult, and they all come out together, right? And then they lay their eggs and then are dormant for 17 years. So it sort of looks like if you only live 2 weeks, not worth it to put in what I assume is a lot of energy to make these beautiful nanopillars. And they also poop all over each other, and I mean, it's craziness. But if you live like an annual cicada, which as an adult can live 2 or 3 months, it is probably worth it to be able to have these structures because you can create super hydrophobic surfaces. I'm going to skip this. I think most of you in the back room probably know what we mean by being super hydrophobic. Basically, that these contact angles are so large because the droplet stays perfectly spherical. So that is what we're seeing in these insects. So we use this really fancy machine in the Milkovich lab that releases these droplets that are identical every time, pretty much, onto a piece, part of the wing that, again, we're pretty sure that it is like, say, the MC cell, and then we measure the contact angles. So we did that for at least 5 different cells on the wing, these different areas. And again, you can see the ones that have these really defined nanopillars are super hydrophobic, whereas the periodical cicada is far less hydrophobic. So that is probably because they don't really need to, because they probably don't really need to be. So they get so super hydrophobic, these annual cicadas, that they start the droplets start jumping, which I guess is something that engineers really want on this material. Like when Nenad saw this, for instance, he was pretty excited. I was excited because look, it's Mickey Mouse right here. Want to see, if you look at Mickey Mouse here, what I mean by jumping droplets is what they start coalescing, and then this makes them jump off of the surface. So the nanopillars are under here. The droplet was resting, or the droplets were resting on it. And so then we also did studies on, can you then actually remove particles? Do cicadas have this mechanism, this function, so they can actually maybe remove particles? And yes, it is that actually does happen. And hopefully this will be published soon. And sometimes you can even just have one big droplet, and it can even jump and remove dirt. So this is what called self-cleaning. So this is really useful for insects, because for cicadas, because you remember cicada wings are sort of translucent. So if they get any dirt on them, they will be a lot more easy to see, let alone if pathogens were growing on them. We also studied antimicrobial activity. And again, this was having these nanopillars made it that microbes did not grow as well. I'm not sure why that is. So either, like model number one is, a surface is so super hydrophobic that no solution can get really close, and also the microbe, the bacteria, can't get close. That would be option number one. And then there's also the other options. This just has been modeled by other groups, but nobody has ever really proven it. Like either the bacterium lands on these nanopillars, and just the nanopillars are like a bed of nails, and then the nails actually go through the cell wall. Or there's just so much stretching that goes on that at certain points the bacterium just like splits apart and ruptures. Or there's actually a buckling and shearing going on. So these are all options. I don't really know how to prove it one way or the other, which way, what is happening. We just know that if there are these nanopillars, bacteria do not like to grow on that surface. Okay, so I hope I showed you that for this multifunctionality that we see in cicada wings, them being super hydrophobic, self-cleaning, antimicrobial, probably having some reasons for affecting a reflectivity, that structure is important. But what about chemistry? These things are made out of something, and they're covered with hydrocarbons and so on, which is often very important in insects. They use hydrocarbons also to communicate with each other or signal each other. It's often not covered with wax and things like that. So we wanted to see if we could actually prove, or to what extent structure is important and what extent chemistry. And this is where then the chemist comes in. So this person, Jessica Roman-Costas, who worked at Searle, she spent months and got a Netflix account, because she would sit there next to the microwave and nuke her sample, which was in a like methanol or something, some kind of solvent, her piece of wing, put it in the microwave for 6 seconds in a tube, and then stop it, release the pressure, put it back, 6 seconds, so on and so on. And she did that for 1 minute, 5 minutes, 20 minutes, something like that. And she had a PhD. That's what she did. But it's really kind of a cool paper. So here's untreated. These nanopillars are straight up, beautifully arranged. After 1 minute, we see these, they're kind of collapsing onto each other. And in the paper, we actually call it kissing. The pillars start kissing each other. So that's after 1 minute. After 2 minutes, it's more kissing, a lot more kissing. You see them coming together. And then after 5 minutes, it's mayhem. Like everything seems to kind of be attached to each other. So as she is removing surface from the nanopillars, they start behaving differently. So what is actually happening is you're making them shorter, and you're also making them thinner. So maybe the aspect ratio changes a little bit. But then after 10 minutes, it's actually coming back. They start standing straight up again, and after 20 minutes, they're almost perfect again. It's just the whole wing is a little bit more papery. It's not as sturdy as the cicada wing that we started out with initially. Okay, and then we did kind of these same kind of tests that we did before. And just to give you the synopsis, if you extract a lot of the surface from the nanopillars, you get this kissing pillar, which should be eye pillars phenomenon, and then that decreases again. Then they start standing straight up again. Hydrophobicity, it's really good initially, but then as they're kissing each other, it's not very hydrophobic, and then it comes back. So this is kind of cool. Like you can actually play with some of these functionalities, and then also antibacterial activity actually increases initially, but then it comes back down. So again, by changing the shapes of these nanopillars, you can actually play with the functionality. All right, so this was actually written up and got some press, and then somebody made this kind of synopsis of a graphical abstract. The cicada, this is what the wings look like. If you the nanopillars on the wings look like. If you put a water droplet on it, it stays like that because it's biophobic. And then if you put Cheerios on it, no, it's actually microbes don't like to grow on this material. Okay, so I just want to, these are the conclusions of it. I just said I just want to focus on this. So if you change the surface chemistry, it affects topography. Yes, chemistry is important. Okay. However, the change in topography also affects functionality, like hydrophobicity, bacterial disease, cleaning. So I think that if you care about hydrophobicity up to a certain level, you can get away with just creating the structure. So you don't have to create, also cover it with chemistry, but we would like to study that. Okay. So if you remember the pie chart, we did biology, we looked at constraints. That's where we're at now. Now we're going to start prototyping. But I'm in a biology lab. I don't have a fancy clean lab access, or definitely don't have the money for it. So usually when we do nano microscale size patterns and features, you do this at a facility like Oak Ridge National Labs. But I didn't want that. I wanted something like my kitchen, which does not look like Oak Ridge National Labs. So can I make this on a biology bench? Can I make materials like cicada wings on the biology bench? So I want a master surface that, in this case, is cicada wings, but we would like to use other insects too, on other parts of interest. Insects. Can we fabricate a mold or the template and fabricate multiple identical replicates? Can we do this over and over again? Can we make big sheets, something? It has to be non-toxic and preferably not have any special equipment. And we were able to do that. Our Jun-Ho Oh in the Milkovich lab was able to do that. However, I do think it was really, really important that all these people are involved in this, and we had lots of meetings together. This was before COVID, and there's different identifying genders in here. And Jun-Ho, I don't think would have figured this one out if he hadn't talked to somebody who regularly gets a manicure. All right, so that's clearly not me, but there are other people in this group. So what he did was, so this is the massive surface, which is the cicada wing in this case. You cover it in nail polish. Seems so simple now, right? But it had to be a special kind of a nail polish. We tried UV ones, the ones where you sit under the thing. We actually have that in the lab. We actually have different kinds of nail polish and the UV light and everything. So we can always become a salon, I guess. So it's hard to see, but then you have a nail polish replica template, and then you either put in a polymer in there, like PDMS or something like that, or actually make a metallic replica, which we can also do. And then you just dissolve that template with a nail polish remover or acetone. So that actually works, and it works very well. Well, maybe my grad student Utah is there now. She's now in charge of making big sheets of it. It's not that easy, and also John Ho sometimes made it sound easier than it really was, but she's figuring it out. So that's what we're up to now. So just to recap, we're using biology to come up with new engineering solutions. So we can now fabricate a cicada inspired material that's super hydrophobic and antimicrobial. And we are using engineering tools, materials to now study biology. Like, how can we now prove that why certain cicadas are a certain way, the way they are? Okay, so I'm gonna. Is it already? Oh, 12. Sorry, it's like I've been talking for an hour, but I think I've been here an hour. Okay. So what we would like to do next is like add chemistry. So this is where I would love to maybe work with the molecule maker lab here at the Beckman to add chemistry back to the surfaces that we are creating. But I guess fatty acid chemistry is actually really hard, and that is actually what we'll be needing to add to the surface. So this is a proposal that I once wrote. I think people liked it, but it's just really hard to make the molecules that we will be needing. So I'll just wait. I'm sure they'll figure it out. Also, we want to study hierarchical structuring. So this is, so we study this in flies. Flies also have wings that are covered with nanopillars, like here, but they also have these microtrichia, these hairs. I don't know if you know this, but insects, when you put them under an SEM, you'll be surprised how hairy they are. There's hairs everywhere, like out of their eyes. There's hairs everywhere. Anyway, these hairs also are structured, and also they affect hydrophobicity. So we're trying to, Utah is trying to figure out these different levels, like what if it's condensations that are happening? Are these nanopillars important? Is the microtrichia important? Those kinds of things. Because as you may now notice, like cicadas are actually really simple. Like they have a simple structure, these nanopillars, and they're nice and sturdy. The wings are nice and sturdy. But flies they're very papery wings, but they have way more structures on them. On the other hand, they also get in some really gross stuff. So they probably really need to be able to stay clean and stay dry. So I think we'll learn a lot from flies. And then Utah also is using these replicates to then put them in the environment and then try to figure out what is actually, can we actually prevent microbes from growing on these surfaces if we make them with the nanostructures that we see in cicadas. Okay, here is another project that came about because I gave another speech to one of the other themes, or what are they called, themes in Beckman? Yeah, was invited by Charles to come give a talk, and then we started talking about what I do, and then he became part of the MURI, and then invited me to join the MURI, which I'm really, really thankful for. That was really great because At the same time, I also started, Elizabeth Bello also started in the lab, and she took on this project. And I mean, this is going to be her PhD. It's great. And thanks to the Beckman for supporting her with a fellowship last year. And then Gabe Bergs, the postdoc in Charles's lab, is also such a great mentor for Lizzie too. So kudos to the Beckman for getting us all together. And we're going to make some really cool stuff because just to introduce you, these are leaf hoppers. They're everywhere. They're really, really tiny. You probably swallow them like a couple every summer. They're pretty varied, but aren't they beautiful? And then Lizzie also won an award for that image called Born This Way. So they come in different kinds and different flavors and different colors. Oops, wrong way. They, cicadas, not cicadas, leaf hoppers cover themselves with brochosomes. These are brochosomes. So they're tiny little particles, about half a micron at the most. These soccer balls, they may have 12 faces, 20 faces, kind of depends on the species. And every single leaf hopper species has them. They make them in their what's called Malpinian tubules, which is kind of like our kidneys, and then excrete them out the back end. And then they use their wings to, I actually have to show you this, like that. They like anoint themselves with these brochosomes all over. And then they might lose them and they make more. So they just completely cover themselves with it. And we think that, well, hardly anybody has studied this, but except for one person, Rakatov, who's now in Russia, but he did his postdoc here at Illinois. Nobody else has really published on these cute little balls. So they're considered give leaf hoppers super hydrophobicity, anti-reflectivity, probably important for surface tensions because these leaf hoppers are so small. Like imagine how they're going to get like off of the surface that might be slightly wet or something. So maybe brochosomes are important. Anti-biofouling probably, and other kind of protection. Really briefly, for a master's, Lizzie just looked at, I think it was 8 different species, 3 different parts of the body, and she measured endlessly these balls, like how big they are, how many faces they have, how many there are, and so on. She compared brochosome size versus species. There is a difference between species, but it doesn't necessarily correlate with the biggest leaf hopper has the biggest brochosomes. Also, brochosome size versus wing location, we thought maybe that would be a difference. As you see, the legs of leaf hoppers have like little teeth on them, and so you could imagine that maybe as it's spreading it over the wings, maybe there's a difference in like maybe bigger ones here and smaller ones there. Turns out that's not the case. There is no difference. There's an effect on wettability on species, and that's probably because brochosome size is different. So this is definitely something that we want to look at further. There will be at least a couple of papers coming out of this with Charles, and then also with his lab and the Chen lab, and we are trying to figure out everything about brochosomes. It's so wide open. So for instance, we looked at nanostructure-derived anti-reflectivity. This is kind of over my head, but this is a lot of, this was recently submitted. So reflectivity changes based on collection of brochosomes, or at least that's what we are modeling it that way. So that this kind of a fun collaboration with the Marianne lab at the University of Texas. This one is with the Chen group here in Beckman. This is mostly Fallon's work, trying to come up with 3D electron tomography reconstruction so we can figure out how much is actually material and what is actually the empty space, and are the differences between species and so on. So this is definitely, I'm bringing this up. This isn't something that doesn't really, I've never seen other entomologists do this kind of stuff. I learned so much, and it's so great appreciation for this kind of work. We also did machine learning from 3D images. I think this will have a lot of application for other kinds of nanostructures that are common in nature. So, but what I did learn is that the data that informs the machine learning is like all that stuff that Lizzie did, and it cannot be less. So like her master's is what was the input here. So, but I think from now on, things will be a lot easier. So if we want to do another 8 species, we can just plug it in over here. So this is really fun collaboration. This is mostly Lihan's work. Okay, another two, we also really would like to know how do these brachiosaurus actually adhere to the surface? How do they stay on? And how do they maybe attach to each other? So we just put in a proposal at the Center of Integrated Nanotechnologies, where we're actually going to use this nano indenter to try and boop and move, let's call it booping the brochosome, or actually moving them and see, like, so see what the elasticity is, or the, yeah, those, that kind of, we don't really know what they're made out of, except for probably lipids and some proteins. So hopefully this will get us closer to figuring that out. All right, so this is still very much beginning. This is why it's still gray. I think we're a little bit more now understanding why it's multifunctional material if you cover with brochosomes, and why not. All right, just a little speech about interdisciplinary research. So all of these ABC lab collaborators, these are all the people that we work with. And we know that interdisciplinary teams and the Beckman, this is well documented, are resilient, effective at decision-making, can be very innovative, very productive, but it is not easy. So we actually work with people that study team science and try to figure out why do some collaborations work and why not, and how can we improve the likelihood that a collaboration might work. So this life in the interdisciplinary boundary, I just want to point out, I think having trust in each other, and also having agreements ahead of time, like understanding that I, as an entomologist, I think having a couple of papers in, I don't know, nanomaterials is great. Sure. But I also every now and then have to publish in an entomology journal, which is a far lower impact factor. But I want to show my colleagues what I learned about insects. So those kinds of agreements you have to kind of come up with at a time. Of course, there's this problem of science silos. How do we talk to each other? Even in biology, entomologists and plant biologists don't even really talk to each other, let alone microbiologists. So anyways, we actually have some money from NSF to study this. And Andy, you probably know Amy, who's now at, I should change this. She's still at Princeton. And then we work with Will Barley in the Department of Communication, so an actual social scientist, to help us understand how we actually work. So we have some money to do that. I think incentives are really important here. The incentive for a biologist to work with an engineer. I mean, it's fun. I mean, I'm married to an engineer. He's pretty fun. I think we can collaborate. But I think from like the students, your student time already. So there needs to be flexibility in academic programs. And this is a real pain for colleges to start sharing instructional units. Whew. And then there needs to be a funding structure and availability. Everybody talks a big game about interdisciplinarity, and here at Beckman probably also have some experience with, but where then do you get the money? Like NSF seems to, well, for a long time I would bump my head. even though I've just came from a workshop from NSF, and they are actually considering to have a whole bio-inspired design track in the future. So that would be great. Of course, then also hiring promotion of when you do interdisciplinary work is tricky. But in the new communication number 25, something like that. So there is here at least at Illinois, there is a recognition of that, which is wonderful. And then reward for non-traditional products. So if I, as an entomologist, actually get a patent, I'm not sure if my department head cares. My department head cares, but that's not necessarily the case. Okay, and just I hope you have an new appreciation for insects. Just want to point out we are losing insects at an alarming rate. Alarming. So for instance, all insects, we've already 41% in the last decade, we've probably lost. And for vertebrates, that's only 22%. Only 22%. So it's worrisome, very worrisome. So it's for all kinds of reasons. Why that is, and how to solve it, we don't really know. But we need to protect biodiversity, insect biodiversity, for moral reasons. Number one, I would argue, important natural functions. Without insects, we don't have chocolate, coffee. What else? Some other really yummy things. Apples, but coffee and chocolate seem most important. Aesthetic reasons. We need continuous evolutionary processes. You can get actually materials like honey and other materials from insects. Okay, bioinspiration, I put it on here. It's really low, I understand. But every time we lose a species, it might have had a different way of solving a problem that we're looking to solve. So that's my soapbox. And I want to thank the Beckman, my department, the university, mechanical engineering for their support, and then all these people from all over the place. Thank you.
That was a fantastic talk. Thank you so much. I think we need another talk on team science. So we'll talk about that. We have a few minutes for questions. Any?
Yeah.
Regarding the cicada project, when you demonstrated that you could strip away the chemistry by heating it in methanol, do you know what chemicals you're actually removing, and if it changed by either cell position or even within each cell?
Can you repeat the question?
Yes. So as we are extracting the chemicals, yes, I didn't show that. She actually did all kinds of chemical GC aspect, all kinds of stuff to figure out what exactly is where. Some stuff that we found is like there's no chitin, which is like the basic molecule in cuticle. It's all hydrocarbons and fatty acids and stuff like that. So yeah, she did all that. We know all that. Yeah.
Thank you.
Mike?
I have a question. Having observed cicadas for decades, there's a difference between annual cicadas and periodical cicadas when they emerge. When the annual cicada emerges, once it's out of its shell and sterilization has begun, it's pretty much happy to go. With periodical cicadas, there's a huge, the major mortality factor occurs between emergence and sterilization. So I'm wondering, is there some developmental thing that leads to this early death, and that might be worth looking at just to see how that it doesn't get to the point where it's resistant enough to bacteria and all these other things. Have you ever looked at non-adults?
I have not, and I think the whole sterilization process, nobody has really looked at that in the last 10, 15 years since Anderson. It's really hard to do. Anytime you mess with
puzzle that why have all these died? They emerge, spend 70 years in the ground, they emerge, spend 20 minutes and die.
yes.
No evolutionary sense.
Yeah. I don't think annual is that fast either. We give them 24 hours to fully spermatize. We don't do it like the next morning. We don't do studies on it. We do it 24 hours. So it's not that fast either. But it also grows vulnerable. The amount of heaps and squirrels is
And I have a structure question. You showed those pillars and they're not actually hexagonal or close packed, they're in spiral patterns. So how much do the patterns matter in terms of the properties that you're looking at? Can you engineer different patterns to get different properties?
definitely would like to be able to do that. Yeah, I'd love to see that. I think the whole, how do you get this pattern and why is it so perfect? And it has to be perfect. And I mean, that is just amazing stuff that I'd love to work with people scientists here, how that happens. Again, it is, cuticle is a fairly simple material, chemistry wise, but then how they get these pillars and they're so perfect. So as they're developing, they're like mush in this nymphal stage. There's a lot of, it's very watery. And then as it dries, and they fluff themselves up. And then you get these perfect wings every time. Well, not every. Yeah, it's yeah.
I think we're out of time. Let's thank Marianne again for everything. Thank you. Yeah, that was